So I think I'll probably just start now. I'll reintroduce myself. I'm Mimoza, and I'm a second-year biomedical science student at St. George's. This is going to be an introduction to neuroanatomy and neuropathology. I hope you enjoy it. I will say some of this stuff is not for the faint-hearted, but I believe it's very, very interesting, and I hope you enjoy it. So, okay, what is the study of the brain? So, as a definition from Wikipedia, it's a multidisciplinary science that combines physiology, anatomy, molecular biology, developmental biology, cytology, which is also cell biology, mathematical modeling, and psychology to understand the fundamental and emergent properties of neurons and neuronal circuits. Basically, words and terms that people use, neuroscience. Literally, as soon as someone says study of the brain, you know they're talking about neuroscience because it encompasses all of these subsections under this one name. So realistically, when people talk about neuroscience, it's kind of divided into these two sections. The first kind of section or branch I would call is neurology, because this involves studying and treating problems associated with the structural, biochemical, and electrical features of the nervous system. Now, this includes neuroanatomy, neurophysiology, and neurochemistry. Now, neurochemistry is to do with how the chemistry of the brain influences mood, arousal, attention, memory. Neurophysiology is to do with identifying the function of different parts of the brain. And neuroanatomy, again, is also to do with this structural idea. So let's say if you had a brain tumor in your frontal part of your skull, then that would obviously have effects on your frontal lobe, and so on and so forth. Stroke, again, if you damage the blood supply to the brain, depending on what part of the brain that you've damaged the blood supply to, you will have consequences to that. Brain atrophy is to do with stuff like Alzheimer's. So it's literally where your brain starts to deteriorate and become smaller in essence, and you lose certain functions and parts of the brain that carry out those functions. The other half to this neuroscience would be psychiatry. So this involves studying and treating problems with mood, behavior, cognition, which is thinking, and how basically you think, and perception, so the way you see things or you understand from your environment, the stimuli around you. And that comes up again under another bunch of branches, so stuff like personality disorders, so experiences and behaviors that differ from social norms and expectations. People like this probably be classified as psychopaths or sociopaths, for example. Severe learning disabilities such as autism, Down syndrome, cerebral palsy, mental illnesses such as basically how it's a difference in how a person behaves, feels, or perceives things and thinks. And then again, there's this idea of whether it should just be called neuropsychiatry. Maybe because I don't know if you guys can see a little bit, but there is kind of this overlap. You know, I know, for example, from the neurochemistry, the influence of mood, arousal, and attention, to basically mental illnesses. So mood is a person's behavior and how they feel towards certain things. And again, personality disorders. So for example, psychopaths don't have the same sympathy as a normal person would. So again, this idea has been toyed around, just maybe calling it neuropsychiatry and not having them as separate things. But yeah, it's not commonly used, but it's an idea that a lot tend to agree with. So this idea, brain versus mind, just want you to wrap your head around what I've just said there. Your brain being your brain and your mind essentially what makes you you, because we all have brains, but our minds are all wired differently. We all think differently. We all act differently. We have different likes and dislikes, have different hobbies, you know. So simply put, your brain is like the hardware inside of a computer, and your mind is the software, because your mind can change and your mind can alter, and you could have like updates in essence. So the more you grow up, the more you understand things differently. But that doesn't mean that there's always a corresponding big dramatical change to your brain, for example, because, you know, we all still have, you know, the front part of our brain, the back part of our brain. It doesn't mean just because your mind has changed that your brain has necessarily changed with it. And unlike a computer, if your brain hardware it becomes destroyed, it's not something that can be easily replaced, not as of yet. So what is neuroanatomy? Basically, anatomy of the nervous system. Now, what is the nervous system? I will give you maybe 5 seconds to think. Maybe, have you done it in school? Do you remember anything? What's the first thing that comes to mind? Nervous system. Well, this is the entire nervous system. I guess this is as deep of a detail as you'll ever get to become familiar with the nervous system. So you've got your central nervous system, which I'm pretty sure you've learned, that's part of your brain and your spinal cord. Then you have your peripheral nervous system, which is basically everything outside the central nervous system. And I'm pretty sure you might have heard of sympathetic and parasympathetic. I guess the biggest shock would be the brain and its gazillion divisions, but I will hopefully get you to understand this partially. So again, what you should probably know, I'm expecting that you know about the brain and the spinal cord. Probably expecting that you know that about the reflex arc. So, your hand feeling the hot flame, sensory input, which there's synapses to the relay neuron, goes up to the brain. Not necessarily if it's a reflex arc, but it then relays to your motor neuron and then causes the effector muscle to react. Synapses, which is basically and yet it's basically transmitting information from one neuron to another. On we go. Now in medicine, we don't call them necessarily sensory and motor. We call them afferents and efferents. So afferents, meaning as in going away from the sensing of what's happened, the heat, the touch, the whatever it's felt, taking this information to the afferent nerve to then be processed and then back via the efferent nerve. So basically your afferent is your sensory neuron and your efferent is your motor neuron. Now, again, we also have another bunch of words that we use to describe this. So your afferent is also your dorsal, which basically means your back. So it's towards the back of this. This is meant to be a vertebral disc, so a spinal cord disc. It's towards the back is your sensory neuron, and towards the front, your ventral, so towards your belly, is your efferent. Now, if we take a look a little bit at the peripheral nervous system, we can see that the first branches are of the autonomic nervous system and of the somatic nervous system. What does this mean? Well, autonomic is involuntary control. So this is control you don't think about. This would be like the reflex arc that I just we talked about. And the somatic is your voluntary control. So this is if you want to, I don't know, get up and walk to the other side of your room, that would be somatic control because you are initiating that response. Autonomic control would be something like you touch something extremely hot, or someone slams the door on your hand, which would hurt a lot. That is the first difference between autonomic and somatic. Now, if we go a bit more into the kind of physiological anatomy aspect, what I don't want you to read all the fancy words on this diagram because you'll be very confused, but this is your somatic nervous system and this is your autonomic. The difference is that your somatic nerves go directly from the central nervous system to the target organ. They do not synapse anywhere. And that would be the first physiological difference from the autonomic system, because the parasympathetic nervous system does synapse somewhere else, and the sympathetic also does synapse somewhere else. Your parasympathetic synapses at a ganglion, which is a bunch of cell bodies close to your target organ. And your sympathetic also synapses at a ganglion, but not so close to the target organ. It's more so close to your spinal cord. Ways of showing this would be this. So this is your spinal cord. And this are in the sympathetic system, where I said that it's that ganglion that it synapses at before it goes to the target organ. So hopefully you know that your parasympathetic, so your PSNS, your parasympathetic nervous system, is your rest and digest system. So this is literally when you are at rest, your parasympathetic system is revving, it's going, it's literally working in your entire body. If something was to happen, like, I don't know, you you're sitting in your bedroom and you hear a bonfire outside, you jump a little bit. That's your sympathetic nervous system going. It's your fight or flight response. So that jumping is literally due to that sound stimuli that your body heard and interpreted, and it made you jump, basically. And then your body analyzes whether it's viable to, is it a situation where I need to fight or I need to flight? So do I need to run away from this? Or maybe, oh, it's just outside, probably just another firework thing. I don't really need to do much about it. So yeah, after that initial jump, your body then starts to analyze the situation, your brain essentially. So again, just to reiterate, this is the ganglion that your sympathetic nervous system synapses at before it goes to the target organ. And then your parasympathetic is this is the ganglions that are closer to the target organs that it synapses at. This is then known, because you've got a lot of sympathetic nerves, this is then known as the paravertebral sympathetic chain. Paravertebral literally just means next to the vertebra. So literally next to the spinal cord. And it's just a chain because all of these ganglia are attached together. So it's nothing so very complicated, hopefully. But yeah, again, you don't have to read all of this and try and memorize it or memorize everything that I'm saying, because it's very, very advanced. Now, if we move to the central nervous system, this is your central nervous system, your brain and your spinal cord. Pretty easy, right? Nothing much more. It gets a bit more complicated than this, in all honesty. So you've got your spine, which is divided into 5 divisions: your cervical, thoracic, lumbar, and sacral and coccygeal nerves. These little squares are meant to represent the bones, so your vertebra in your spine. And the way that this works, so you see you've got C1, C3, C4, C5, C6. So they basically named cervical first nerve, cervical fourth nerve, cervical fifth nerve, and that's literally the way it works down. Again here, thoracic first nerve, thoracic second nerve. But no one literally says thoracic second nerve because that's quite long, so we just say T2. There's 8 cervical nerves, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal normally, but there are variations up until 3 coccygeal nerves. So that we call those anatomical variations. So because not every single person is the same, we've got a couple of variants, differences. It doesn't mean a lot, it's just the way it's found. So yeah. There are cranial nerves here that are not shown. And these literally would be, because this is your your cervical part of your spine. Up here would be your brainstem, and out of your brainstem would come out these cranial nerves. So there is 12 cranial nerves. Pretty much all of them have been shown here. You don't have to memorize these, because if you did that would be amazing, but they are quite hard. And they've all got their own functions. So for example, your optic is your second cranial nerve, and that's to do with your eyesight. Olfactory is this one here is your olfactory bulb and tract, which is to do with your smell, for example. But yeah, that's a bunch of cranial nerves. This is them on a real brain. So this is an inferior aspect of your brain. So imagine looking up into your head, or up into someone's head from below. So the inferior aspect of the brain. And here we can see, basically, these are very clear. I was even surprised when I found this myself. But yeah, it's a nice example. These would be arteries, by the way. That would not be the nerves, but these are the nerves. As you can see, they also vary in size. And yeah, it can be mainly due to function as well. But yeah. So in your spine, then, if we were to integrate what the actual vertebral bone looks like with the spinal cord, they look a bit like this. So your spinal cord comes after your vertebral disc. So it's bone, spinal cord, bone. These are like your spinal processes. And I don't know if you could feel your back, but sometimes people bend over. I call them like dinosaur scales, and literally like you can, or dinosaur bumps, sorry. You can feel like bumps or part on your spine, and then that's how you know to orientate yourself always, is that this is your back part and that's your front part. So this is your ventral part of the back, and this is your dorsal part. This is again a bit more zoomed into the spinal cord. So remember, this was your dorsal pathway, your afferent, and this is your ventral, your efferent. So your efferent was your motor, and your dorsal was your sensory. Yes. This we're going to look into a bit more now, this little bump, but it comes in a bit. Your spinal cord is covered in layers. These are the layers. Your pia mater is the one that completely hugs the spinal cord. Then you've got your arachnoid mater, which doesn't completely hug it, because you've got space here for stuff like lymphatics, blood vessels, your veins, and more nerves. And this space in here would be called the subarachnoid space. So sub meaning just below. And on top of the arachnoid mater, you've got the Dura Mater. And also one thing also to point out is with your spinal cord, you've got your white matter on the outside and your gray matter on the inside. And with the brain, it's literally the opposite way around. And we'll discuss in a bit what white matter and gray matter are. Again, another image. So this would be the nerves leaving your spinal cord. They haven't filled these ones in. It's not like they're just very short. It's just they haven't filled them in. And again, this is just a spiny process that I was talking about that comes out from your vertebra, and it gives you that dinosaur bumps on your back. It's your vertebra, it's your spinal cord. And you see that your dorsal and your ventral nerves here. Okay, again, the vision, what we just learned. So your dorsal root, remember this was the information that's just come in, your sensory information, trying to find out what the stimulus is. It's going in like this. Synapses, and comes out of your ventral root. Your ventral root is then going to go to your effector muscle and cause the reflex. Then on this kind of part, you've got this white ramus and this gray ramus, which allows the connection of, if you remember, the autonomic system, so your parasympathetic and your sympathetic. So those ganglia, remember, because the sympathetic, they synapse at ganglia very close to the spinal cord. They would be connected by these. Again, just trying to show you from now we're looking down into someone's spinal cord, so you've got a superior view from this aspect. This is your I would say ignore the stimulation lead because I did take this from a paper, but just I want you to see that again. You can orientate because you can say this is the back, this is the dorsal part of the vertebra, and this is the front part, so the ventral part towards your belly. Now you can see that this would be the white and the gray ramus, and they connect to the sympathetic ganglion. So that was your autonomic system. The involuntary part. Again, now I just want to bring it a bit more into life so you can kind of really understand. We've discussed all of this, and then this is your paravertebral chain. Because I feel like, in my personal experience, people don't really do justice in teaching this properly. They just say, you know, this is the spinal cord. There's also a paravertebral chain that exists, and that's it. They don't really explain how they're connected. So thought I'd do this myself and just say that it's good to appreciate the integration of the central nervous system and the peripheral nervous system, and then the involuntary peripheral control, which is your parasympathetic and your sympathetic, which is done by this. Remember, the paravertebral one, the ganglia next to the spinal cord, is for the sympathetic, and the one that's closer to the target organ is for the parasympathetic. If you can notice, you see that they're using the terminology T1 to L1, L2, L4, L5. They're telling you what part of the spinal cord that this is taking place. Again, don't worry about this. It's just taken from a paper, and they were trying to determine a procedure. Another image. So again, you can just see the ganglia. The red ones, by the way, are the sympathetic, and the blue ones are the parasympathetic. And literally, this was just to show the relation of the white ramus and the gray ramus being connected to this. This is what it looks like in real life. So it doesn't really look anything like the pictures, because remember, in a lot of this stuff is just like to give an example and an understanding of, you know, this is the function that it has, but doesn't mean it looks exactly like this. And then that's what I like about being able to have time with essentially dissection room time, which is where you spend time with dead bodies in the university, because then you really get to see that, okay, even though I've always been taught that, it doesn't actually look like this, it looks like that. But it's just getting that idea in your head that the ganglia are then connected by branches, and this is where they would be. And the ganglia all do have names. Don't have to worry about that now. But based on the names on this image, this is your abdomen. So this is past your diaphragm and all the way down here, which is this is where your arteries bifurcate to go down to your legs. So this is bifurcation, just means dividing into two. And yeah, that's how you know. If we go up a bit into your thoracic cavity, which is your lungs, your heart, all of that, you can see it here. And this is actually quite deeply entrenched into muscle. So it's not something that you would visibly see. This is between two sheets of muscle. So you can see this in the intercostal muscles. You can see the rib cage, right? And it's in between the intercostal muscles, which is where the blood vessels and the nerves and the veins are found. Okay, so some background, just, you know, getting some background information about how this works. The brain can be cut in different ways, and then that tells us different things. So let's say you wanted to identify a brain region you'd have to be able to identify it on a coronal plane, a sagittal plane, and a horizontal plane. The way to remember these planes is that your horizontal plane is, imagine like an axis, the horizontal axis, the x-axis. And yeah, so literally the horizon, for example. Your coronal section is the way you would wear a crown. So literally, it's cutting the brain this way. And your sagittal section, imagine holding a bow and arrow, and the direction in which you hold your bow is, and that's how you know your sagittal sections are taken this way. Another bit more of brain terminology. Your posterior part of the brain is the back. Anterior, the front. Inferior means as in below, and your superior is the top. Now, the way that we came up with this terminology was not because of studying human brains. We actually, and we still do, study rat brains. And the idea behind it was that we'd only have to call them rostral and caudal, because the rostral is at the head and the caudal was at the tail. But the thing is, with our nervous system, unlike the rats, it's not straight, it's not in one plane. It actually tilts and kind of almost does a 90 degrees down. So when people talk about caudal brain regions or caudal organs, they mean as in away from the head, down your spine, pardon me. So anything that's rostral, you know, is towards the front of the nose, for example. And anything that's caudal is towards the back and then down your spine. Dorsal and ventral, again, we describe this. So your dorsal is anything that's towards your back, and ventral, anything towards your belly, for example. Now we can start going through crazy divisions of the brain. These fancy names down here are further divisions of these names. Now, these names come from the actual embryology of the brain. So we study embryos and we see how actually we go from this many week old to this many week old to actually almost giving birth. So you can see the names here. I mean, I don't want you to read anything off here, literally just appreciate that this is where all the names are coming from. So the telencephalon is this dark purplish part, bluey. Diencephalon is here. Your mesencephalon is here. Your metencephalon is here. Myelencephalon here, and then this becomes your spine. And that was all of that. A real brain. This has been taken as a sagittal view. So remember, bow and arrow goes straight down across the middle of your face. And I guess I could roughly show you where the encephalons are. But this is your cerebral cortex. So all of this bit. This would be white matter. So we'll go on to that in a bit. It's called corpus callosum. But this, essentially all of this bit up here is your telencephalon. This middle bit here is your diencephalon. Your midbrain is your metencephalon. This part is your myelencephalon. I think I've just confused that. Mesencephalon, sorry. Mesencephalon, metencephalon, myelencephalon. Here they're in a nicer drawing, so you can see that a bit better. These are really old names, if I'm very honest. We do still use them, but normally when you're referring to pathologies or problems present in the patient, they will just be like really straight up and just say, oh, yeah, they've got a problem with their hippocampus or their amygdala, or they'll be very like specific about it. They won't say, oh yeah, but there's a problem in the diencephalon, because it's describing a region and there's many stuff inside of that region. So it's kind of just you're grouping certain things in the brain into different groups, and that doesn't really help when it gives you a rough idea, but it doesn't really help when you're trying to be specific. So what is the nervous system made up of? I guess I'll give you 10 seconds to think. What do you know? Like, what's been said to you that you've been told, I don't know, that the nervous system is made up of neurons? This shouldn't come as a big surprise to people, considering you guys have all sat your GCSEs, hopefully, and passed and done well. But there's another guy part of this. It's called glia. This term is not really thrown around, and it's because the glia are more present in the brain. They're basically like neurons, but they're not. And they're neurons specific to the brain. So yeah, what is the difference? Well, neurons have axons and dendrites, and they can generate nerve impulses or action potentials. And glia, they're non-neuronal, so they can't generate action potentials. In actual fact, all they do is just regulate. Because hopefully you guys know what neurotransmitters are. It's the chemicals that cause communication between one neuron and another. And now when that neurotransmitter has to be regulated, like you can't have loads of neurotransmitter in a neurosynaptic cleft, because you will then end up stimulating the post-synaptic neuron a lot more than you need to. For example, like imagine you just wanted to, blink your eyes because of a bright light you saw like a couple times, but then if your neurons keep firing, you're going to keep blinking repeatedly for a very long period of time, and it's just like you don't want to, but because you've got such a humongous amount of neurotransmitter there, it's just going to keep causing you, it's stimulating the other post neuron, and yeah, you'll be blinking like crazy. Yeah, so the white and gray matter are made up by these neurons in glia. Yeah, so now neurons, I guess you guys have always drawn them in one way, and it's probably this one. But not all neurons actually look like this. I wouldn't say you've been lied to, you just haven't been given the full picture, I guess. You've got different types. You've probably learned about this when doing the eye, the bipolar nerves. And it's one of the cell bodies in the middle. And your pseudo-unipolar nerves have got no wrapping, none of this myelin sheath going across here. And your unipolar means that they can only stimulate from one direction, in one side. So they haven't really, it's just the cell body, haven't got any dendrites over here. So yeah. In the brain, again, they're not really called neurons, more like glia. There's different types of neuroglia. They all do different jobs. In your central nervous system, so I'll just give a brief understanding of these. So your oligodendrocytes, they produce the myelin sheath for the neurons. Your ependymal cells, produce CSF, so cerebral spinal fluid, because I don't know if you guys knew, but your brain is literally swimming in cerebral spinal fluid. It's very delicate, so it needs to be cushioned in a way, and that cerebral spinal fluid does that. Your microglia are to do with immunity, and astrocytes are again to have that job of regulating the action potentials going on in your brain, and they help with memory formation as well. Your peripheral nervous system, Schwann cells, again, the myelin sheath business, and satellite cells have a similar kind of job. The way that the cells are organized in the brain, pretty much like this. It's not a very neat organization. There's many layers to the brain as well. So you've got your normal brain is called, it's got neocortex in it, so it's got about 6 layers of cells, different cells, and it can vary up to 3 layers, mesocortex, allocortex, which has only got up to 3 layers, but that again depends on brain region. What is gray matter and what is white matter? Well, simply put, your gray matter is that cell body. So literally, you go back here, it's literally this. Your gray matter is made up of these cell bodies, essentially, or maybe using the neuroglia, these cell bodies. And your white matter is your axons. your gray matter does more of the processing kind of job in your brain, and your white matter literally just gets the information there. It's a mode of transport. So let's say you had a piece of information that came from this part of the brain and it wanted to get to here, well then cell bodies would send the information through the white tract and to get to here. So then basically communicate with the cell bodies in this area of the brain. Or it can go from here and then pass this piece and go to the other side of the brain as well. Could go from different lobes completely, go through there as well. But yeah, you got a bunch of loads of connections. I think there's about, like, estimated currently, as what I know, about 100 trillion connections and 100 billion neurons in your brain. So you've got quite a lot. Where is the brain kept safe? Well, your skull. Where else? Again, don't read the names. There's a lot coming for you in medical school, I'll tell you that much. But your brain essentially is kept in this bit. Yeah, that's your brain. It's actually quite close to your eyes, more close to your eyes than people actually anticipate. It comes, this is again from an inferior point of view, so looking up into your head, literally. It comes out of this hole known as foramen magnum, and this is where your spinal cord comes out of. These other bunch of holes is where your cranial nerves come out of, and where your arteries into the head to give blood supply. Here they've actually removed the jaw, if you couldn't tell, because you can only see the roof of your mouth with the as part of your teeth. So yeah. Oh, and also one thing I find quite interesting that for some reason was never made aware to me prior to med school was that your bone is completely cartilage, so you won't see a bone on the skull because it's not bone. No, sorry, on the skull because it's not bone. Okay, so now I want you to imagine, let's say now, I don't know, you took a pen and we were going to, you had this, say this play skull or this play head, obviously it'd be dissected head, we're actually doing this to real humans, and you were now going to pierce the head of this preserved head, and we're just going to talk about all the layers that we're actually piercing through. Now, one of the mnemonics that we learn is scalp. So skin, connective tissue, aponeurosis, low areolar tissue, and pericranium or bone. Your scalp, your skin, sorry, obviously is your skin with your hair and everything. Your connective tissue is your like collagen stuff under your skin, which gives it its stretchiness, or your elastin. Your aponeurosis is all the neuronal connections, which then obviously come under your connective tissue. So obviously, you know, you've still got a sense of touch on your scalp, so you can still feel temperature, pain, and all that stuff. So obviously you've got neuronal connections there. You've got loose areolar tissue, which basically means your blood works, all in this layer. Or I can show them here as well. Yeah. And then your peristynium or pericranium, basically the top part of your bone, because this is sometimes manipulated, but yeah. That would be the last layer before we get to the bone, or basically the bone. Okay, now we've got to the bone. A couple more layers to go. Past your skull, you've got another layer known as your Dura Mater. Now, the reason why you've got two divisions here, your peristeal and your meningeal, your peristeal is the one that's closer to the peristenium, and your meningeal is the one that is closer to your arachnoid. And that's literally the only difference. But if you were to dissect a skull and remove it off a human dissection, this Dura Mater would come with the skull. It wouldn't remain on the skin, on the bone, sorry, on the brain. Oh. Then you've got this other layer, this white layer here, which is known as your arachnoid layer. So remember, it's again like the one with the spinal cord. Remember, you had your pia, your arachnoid, and your dura. So again, the same casings: dura, arachnoid, pia, arachnoid, dura. Again, you've got your subarachnoid space, Here you've got an extra space, subdural space, which is basically meaning under the dura. Anything sub means under. And yeah, so your subarachnoid space, and then your pia mater, which hugs the brain, your cerebral cortex. And now we have finally reached the brain. So this is actually what a fresh brain looks like. This was, I think, came out of a freshly dissected body of an old man. I got this off a YouTube video. I think it was pretty cool. This, because it's such a fresh brain and it hasn't been fixed, if you actually held it in your hand, it would feel almost jelly-like, and it's quite wobbly as well because it's full of blood and probably some cerebral spinal fluid inside in your ventricles. But you can see that, let's say this woman, she kept that in her hand for, I don't know, 5 minutes. The brain would actually start to mold around her fingers because the brain is really that delicate. That's why it's so important to have it cushioned in cerebral spinal fluid, because your brain can actually is very, very susceptible to damage. And the reason why it has all those layers of protection, because again, very susceptible to damage. Even the skull itself is actually kind of molded to really, like, it's like a perfect lock and key, like a perfect kind of modeled around the brain. I think it's very, very beautiful. Like if you were to look down into an open skull, you'd see loads of notches and cavities, which is pretty much perfect fit for the brain. The ones that we use in the dissection rooms would look a bit like this. We basically, what's done is you pump out all the blood of this brain, you pump in loads of formaldehyde, and then you flash freeze it at a very low temperature for about 1 to 2 days. And then it's basically pretty much ready to use and to do research on. We use this to identify structures, anatomical variants, and so on and so forth. If you're in a lab, however, like let's say you did a test on, I don't know, you drugged loads of rats with cocaine and you wanted to see what effect that had on the brain, then you would actually flash freeze, do the whole same process again, and then you would actually set it in agar or paraffin wax, and you would cut them into extremely thin slices and look under a microscope to see what the cellular structure, what's happened basically to the cellular structure. So what are the weird bumps and grooves on the brain called? Well, any bump is known as a gyrus, many gyri, and any groove or indentation is known as a sulcus or sulci. Sulci being plural. So gyrus, sulcus, gyrus, sulcus, gyrus, sulcus, gyrus. Okay, let's move on to some divisions. Let's really get into the brain now. So this is a lateral aspect of the brain. So we're looking at the outside. Let's say you just look down into someone's head, or just look in from the ear, for example, looking at their ear and into their brain, you would see this. So this part of the brain is known as the frontal lobe. And this is divided by the central sulcus and to make the parietal lobe. You then have the lateral sylvian fissure, which then divides the parietal and the frontal from the temporal lobe. And then you have the parietal occipital fissure here, and an imaginary line that comes right down here to the preoccipital notch. There's not really a set sulcus, in essence, that divides the temporal lobe and the occipital lobe. Now, if we were to cut the brain, again, mid-sagittal, so imagine the bow and arrow. Right down the middle, you see your frontal lobe. Central sulcus has just come down here. This is now a new sulcus, this one here, the singular sulcus, which then flicks up to become pars marginalis here. That then divides the parietal lobe from the frontal lobe. Central sulcus, pars marginalis, this line here, and singular sulcus. This is then your parietal lobe divided by the parietal occipital sulcus. It's the occipital lobe. Again, I know it's a lot of new words, but I promise you, I just want you to understand the lobes and what divides them. So frontal lobe, central sulcus. Remember, frontal lobe, central sulcus. The parietal lobe is then divided by the central sulcus from the frontal lobe. Divisions from the frontal lobe and the parietal, your lateral fissure or your Sylvian fissure. The temporal lobe, that's the division. And an imaginary line comes down to the preoccipital notch to divide the temporal from the occipital. And that's about it. I mean, if you can learn the parietal occipital sulcus, which is about here, and comes in down there. And pretty much, yeah. Yeah. So your brain is divided into two. Now, this is a superior aspect of the brain, so a top view or dorsal view, because it's in. If I showed you that picture again of when I was talking about the mice from the head and the tail. So I said anything towards the front was rostral, anything towards the back was caudal, and then you had the line where on top of the line was written dorsal, and below the line was written ventral. Ventral literally just means closer to your belly or closer with the front of your body. Dorsal, anything away from your body. So yeah, dorsal or lateral or superior is perfectly fine used to describe this. You've got a longitudinal fissure, which is basically this, that causes the divisions between the two hemispheres. You've got your left hemisphere and your right hemisphere. Here we've got a nice representation of the central sulcus on the left and a nice representation on the right. Normally, we tend to use left-sided brains because the fissures and the sulci look a lot nicer and a lot clearer, and the gyri don't look so messy. So we tend to use a lot of left hemispheres. But yeah, so anything in front of the central sulcus is frontal lobe, anything behind is parietal lobe, and down here, your little occipital lobe, which you can see here as well. Now, if I was to take this brain and not in a very violent way, start ripping off the top bits of white matter, because the way that you do brain dissections is, it's not like very nice, like I'm going to get a scalp and, you know, let's say if you're doing a heart or if you're doing a liver and just slowly cut this bit off and then I know that this is here. The brain is really not like that. You could literally take a spoon and just scrape it, because the connections allow you to do that. The way the brain is allows you to do that. So for example, if I wanted to map a tract or basically map a pathway at which the neurons take, so let's say this pathway would literally be information coming from the left hemisphere to go into the right hemisphere, and that would be via this thing called the corpus callosum, which causes the connections from the left side of the brain to the right side of the brain. So if I wanted to do something on this, if I got information up here in my brain and I wanted, part of the processing is that the other side of the brain also has to know to understand what's going on. Then let's say I was trying to pick up a box, I would need, I know it's a pretty big box, I wouldn't just use my left hand to pick up the box, I'd have to use both of my hands, and my brain would understand that, because I'm not only talking to this side of the brain, I'm also talking to the other side of the brain. And one half of the brain controls one half of the body, another half controls the other half. And they communicate with each other via this corpus callosum. So, I probably should have mentioned as well, your right side of the brain actually controls your left side of the body, and your left side of the brain controls your right side of the body. And this is because of something funny that happens in your brainstem. So literally what happens is all the information from here, it goes down into your brainstem and it does something called decussates. So it literally just jumps over to the other side. So let's say the information came down from your left side of the brain, would go into your brainstem and then jump to the other side and go and just flow down the right side of your body. So to control your right hand, you would use your left brain, and to control your left hand, you would use your right brain. Left versus right. You've probably heard this. It became a famous theory not too far back in the day, but it's not really supported, but it's not at the same time really like disliked or disproved yet. the argument is that your left brain does more of the serious, factual kind of things. So all your reading and all the stuff that you do, that's just, you know, none of all that wishy-washy stuff like, oh, what color is the carpet, or what kind of light color am I seeing right now. It's just literally that, like, I'm reading a paragraph about cats. That's it. They don't care about, you know, the font or the color of the page, just cats. That's what I'm reading. And then your right brain kind of focuses more on the other things the left brain kind of misses out on, which is your, oh, what color or what font? Is anyone reading it? What does their voice sound like? All of the other kind of the accessory parts of that information, of that stimuli that you're looking at. Whether a left brainer reads sitting up or lying down, it's not been proven. I'm not even sure why it exists. And whether left brain people are more dog lovers than cat lovers, I don't think whether you're left brained or right brained really matters on that either. But this professor from Harvard did say the notions of left and right brainness are widespread and widely accepted, but they may also be wrong. So just because basically something is been talked about a lot or been really popularized does not mean that, oh, it's actually right. So the best way of explaining is this: the idea that some brain functions reside more on one side of the brain than the other is true. But then this really depends on the function. Because like I said, the motor and sensory function, so what you feel, let's say you touch something and you felt the temperature, or you felt pain on someone, you know, squeezing your hand, or, you know, fine touch, feeling different textures, or crude touch, you know, being able to understand that there's a corner here or there's a smooth surface. That, for example, your left leg and left arm would be all dealt by your right brain, and vice versa. But something like language, understanding, you know, because the way that your brain understands language is converting text into audio, this is actually generally found more on your left brain rather than your right brain. It's not to mean that if you damage this area in your brain on the left, on your right, sorry, that you would not be able to understand any language. But if it was damaged on your left, because that's where the language is based, then you would have more problems. So if you had the same lesion on your right, you would still have problems, but they wouldn't be as severe as if you had that same area damaged on your left side, for language, for example. Now, how do we study the brain? Because we wouldn't know all this stuff because it wasn't just kind of spoon-fed to us. People had to do real, you know, staying up late at work, coming up with crazy ideas and, you know, investing loads of money into it to try and understand what the brain is. And we've only been doing study of brain for about the last 50 years, actually. And probably a tiny bit more, but that's about it. You know, there is so much we don't know compared to what we do know. And yeah, so hopefully we've got a lot of potential neuroscience scientists out there ready and gearing to go to do study in brain. But basically, we use mice and rats. And the way that we do this, we don't just take any odd rat off the street because we don't know what kind of life they've led, you know, what they eat, what kind of genes they have, or how susceptible they are to certain diseases. So we basically breed them ourselves. We genetically make them ourselves, breed them, use them for experiments, and then sadly, we have to kill them because we cannot release these rats into the wild, or mice into the wild, to which then could then disturb the ecosystems currently that we have in place. This mice right now has got a device in its head trying to, I think, identify brain activity. So, sorry, understanding what's going on in there, but I won't go into too much detail. This is actually how we harvest brains, or how we cause lesions to brains to then see what the consequences of that are. And this is a fancy machine we use. This rat is either dead or alive and anesthetized, so they won't feel any of this. And your brain doesn't have any brain receptors either, so more of the pain would be coming from actually slicing them open rather than actually pointing this item into the brain. And this is done with all specific coordinates because we have mapped out the brain like that. So yeah, We also do from normal pathologies of living people. So we do research studies. So this is a brain cancer, Geoblastoma, and this is shown on an MRI scan. This patient has got a very wild, very huge cancer. It looks quite huge on here, but you can see here it's got a bit of a shadow. This is edema, so this is the water that's accumulated around the tumor, but the tumor is only actually that big. But yeah, you can see that it's squashed a lot of this side of the brain, and that person is probably suffering a lot from it. Or we just do a post-mortem study. So we harvest brains from diseased patients or patients that have died due to disease. And we basically look at their brains and just see, this is what a normal brain would look like. And this is, again, brain atrophy, Alzheimer's patients. So they've got an absolute complete shrinkage of their hippocampus, because your hippocampus is to do with forming memories. And a very big symptom, I guess, of Alzheimer's is the fact that you can't remember things, or you're very forgetful. So yeah, and you can see that here, extreme shrinkage, so a lot of atrophy there. So what do the different parts of the brain do? I won't go too much into this. It's literally what it says on the page. Your frontal lobe, look at this. There's a lot. This would be called your prefrontal, but this in its entirety would be called your frontal. So your prefrontal does a lot of the attention, planning, decision making. Your frontal, this part after the prefrontal, does a lot of your movement. So if you just want to move things with your hand or anything, your touch and feel and sensory aspect comes into the parietal part. This is also to do with your balance as well. Your occipital lobe to do with vision, and your temporal lobe is to do with smell, memory processing, and emotion. This is your cerebellum, which is to do basically with literally coordinating movement, because we haven't done a lot of studies into it. That, as of right now, this is the only feature that we know it has. Your brainstem is to do with more of the parts you don't think about. So your breathing and your heart rate. So in old textbooks, this was called as the old brain, because all species have this, all mammals have this, all hot-blooded animals have this. And then the newer brains formed because of these loads of this new formation of gyri and sulci. Basically, the more information or the more complex of an organism that you are, the more gyri and sulci that you actually have. So in a cat's brain, this would be completely smooth, because they don't need so much, I don't know, gyri or so much information or ability to make decisions, or, it's what really makes us more complex as humans compared to other animals, is the fact that our brain is more complex, more developed. So the more information, the more developed, the more gyri and sulci, the more lines and bumps and grooves that you'll have. So there are other systems present along the brain. Again, I will not go over these, but you've got your vasculature, so your brain, your water, your blood supply to your brain, which is this. So the brain specifically, this one's in a circle of Willis. You've got your venous drainage. So obviously, after all your cells have loads of lovely glucose and oxygen, they've produced lots of now not so helpful CO2 and water. So that is actually drained by your sinuses, your venous drainage, which then goes back into your heart and lungs. Your ventricular system, this is your cerebral spinal fluid. So as I said, it was literally your brain swimming in this fluid, and this is that fluid. You can see here, it's all that lovely light blue fluid going everywhere, and it actually just goes all the way down your spine as well. Your spinal cord is drenched in this stuff as well. And inside, you've actually got some space inside of your brain, and it looks a little bit like this. And this is the inside, or the inside portion of the ventricular system inside your brain. So this, each wing, these are the wings, one's on the left side, one's on the right side, and they coalesce here. Third ventricle, fourth ventricle, and they leave. If we had time at the end, we could do this, I guess. So what is neuropathology? Neuropathology is the study of disease of the nervous system tissue, usually in the form of either small surgical biopsies or whole body autopsies. So biopsies you take when the patient is alive, so they can go into surgery. Let's say you wanted to test their brain tissue, you'd go into surgery, take a bit of their brain tissue and study it, but the patient would still be alive. You stitch them up, and go and carry on with their normal lives. Whole body autopsies, obviously when the body is dead. So if patients died due to Alzheimer's, you can take their autopsy and study them, I guess. Again, not for the faint-hearted. So I have really started with one of my favorite procedures. Not to do, but to read about, because I thought this was actually really interesting. I don't know why it's not talked about more. But yeah. So this guy called Is Neuroanatomy, he was a Portuguese neurosurgeon, and created this procedure known as the leucotomy, or the prefrontal lobotomy. Then it was developed further by Dr. Walter Freeman and James Watts, and they started doing more in the US, and Walter Freeman was really the one who tried to popularize this technique. Igaz was more of the person who said, you know, I'm going to create this, but I'm only going to use it when it's absolutely necessary. Walter Freeman, on the other hand, you know, saw every person as a potential person who could have this prefrontal lobotomy, which is a bit crazy actually, because if you didn't need it and you had it, you suffered very severe consequences from it. So the claim behind this was that it permanently reduced violent, maniac, and psychotic behaviors in patients. And Igaz specifically said that he would only use this when all other options have failed. Now, what this really does is a form of therapeutic nihilism. So this basically makes you very toned down as a person. It cuts all the connections from your prefrontal cortex, not maybe all of them, but it cuts the prefrontal cortex connections to the thalamus, which is more deep inside your brain. Prefrontal, remember, was this part, your attention, your planning, your decision making. I think someone's accidentally drawn on my my graph, but that's my slide, but that's fine. So this is the procedure. You basically take this instrument here and you stick it up someone's eye. And because I said your eye is very close to your brain, you're going to go in here and you're just going to wiggle it around. You take it back out, you put it in a bit deeper up here, you wiggle it again, you take it out, one more time, goes back in, and you wiggle it again, and then you take it out. But you have to put it at very precise angles and only at specific depths, because if you go one step too far, we're going to go into the movement part of your brain, which is then not very helpful if you're touching the movement neurons, because then basically you can't move. Again, another picture. It looks very horrible. I mean, there's no other way to put it. This did cause a lot of problems, like I said. Some of the problems was the fact that they had to be taught to eat and use the bathroom again. Again, like I said, if you go too far, you can't use your limbs. Some people actually became permanently childlike, and others were actually in a vegetative state, which basically means you are in like a coma. You can't talk, you can't eat. You're just, your heart's beating, you're just alive, but you're not really alive. And then people would either die from this procedure or have a relapse. So the violent maniac and psychotic behaviors would come back. This is what people essentially look like. I mean, it would go from really happy to really not so happy. What this is really just trying to get is that you become very subtle. You don't have or express these emotions, these things that essentially make you human. Because out of a lot, if you do comparative anatomy, so if you compare anatomy from our brains to other animal brains, you would see that we've actually got the largest prefrontal cortex, and we think that this is what makes us more human than others, is the fact that we can make decisions, we can think about things, we can make goals and try to achieve those goals, and there's things that we look forward to and there's things that we don't look forward to, you know, all these emotions, these characteristics that make you human. You're kind of disturbing those connections and making yourself just alive, I guess. And there's people, I mean, who've said, I'd rather die and get myself shot and killed rather than go through a lobotomy, because they're absolutely horrible. So, moving on to another story. Phineas Gage, he was, basically this is what happened to him, and he actually managed to survive, crazily enough. He was a railway worker, in 1848, and Gage used a tamping iron to pack in gunpowder for blasting a tunnel through a rock. While his head was slightly turned, a mistaken strike sparked an explosion that forced the rod upwards into his left eye and out through his skull. Miraculously, Gage survived, blinded in his left eye, and sustained damage to much of his left frontal lobe. After the accident, others noticed changes in Gage's personality. Before the accident, he was known as a responsible, hardworking, but then afterwards, again, complete change in character. He became disrespectful, foul-mouthed, and had difficulty carrying plans out. So again, this pathology, this damage, helped us understand really what that part of the brain does. Another pathology. This comes in two forms. So first, this used to be called the Arnold Chiari malformation. There's type 1, type 2. And basically what happens is that your cerebellum is kind of getting sucked out of your skull and into your spine, actually, funnily enough. This can be congenital, so you can be born with this, or it can be caused by trauma, I believe. Correct me if I'm wrong. But basically what happens is your cerebellum starts doing this, and you start having horrible headaches, and you lose a lot of your balance. Or it can also be caused due to inflammation as well because of hydrocephalus. So hydrocephalus issue just means that your ventricles are very inflated because you've got loads of CSF pumping around, and there's no space to actually keep the cerebellum in there, so it starts trying to go where there is space, which apparently is the spinal cord. It's quite horrible. You have to get surgery to fix this. It's very uncomfortable to live with. People who have Chiari 1 actually malformation tend to be able to live with it to some extent, but again, depending on how severe it is, there are actually more subsections of this. There's Chiari 3, 4, and 5. So again, depending on how bad it is, you will actually have to get it treated. Prosopagnosia. So this is actually facial blindness. So prosopon, face, agnosia, not knowing. You literally cannot recognize people's face because the way that your brain is programmed is to see 2 eyes and a mouth and associate that with a face. Now, when your brain is seeing 2 eyes and a mouth but not carrying out that association aspect, it's seeing a face, but it doesn't know who it is. And it's quite sad, in all honesty, because what can happen is people don't tend to recognize their own reflection, or they don't tend to recognize people whom they actually love and care about. So, for example, you wouldn't be able to recognize your mum or your dad by face. These patients rely on other cues like sound or touch to recognize who that person is. So let's say you only recognize your own mum due to the fact of her voice, not of her face, because your brain does not have the ability to make that association. Normally what's affected is the fusiform gyrus. Again, an inferior aspect of the brain would be this. Fusiform because it's meant to look a little bit like a conical flask or like a little tube for some reason. I don't see it, honestly, but yeah, that's the part of the brain which has actually got a problem. Again, sagittal view, think about the bow and arrow. Looking at the bottom bit here in the temporal lobe, fusiform gyrus, again, that's what's been damaged. Again, damage from anything, tumor, stroke, reduced blood supply to that area would cause this, or it can be congenital where you're born with this. famous person born with this, or technically you treated a patient with this, was neurologist Oliver Sacks. And it was that bad that the man mistook his wife for a hat. So I guess it can really depend. It's interesting, I guess. A person with prosopagnosia would not be able to recognize that all of these four faces are different. And I hope that you can recognize that they're all different because of the eyes that have been photoshopped into this image. So this has got lighter eyes, different eyes again. These eyes are closer to each other, they're further apart. But a person with prosopagnosia would say that all four pictures are the same person. Hemispatial neglect. Now this is another interesting one. So if you damage your left hemisphere, you can't see the right side of space, and vice versa. Right hemispheric damage is more common. So it's like saying, okay, if I damage the left side of my brain, which controls the movement of my hand and of my arm and my leg, then that means I'll be paralyzed on that part. So I'd have hemiparalysis. This damage again is caused to stroke. Temporoparietal junction literally just means where the temporal and parietal lobes meet. Posterior parietal cortex just means the back part of the parietal lobe. What happens is, so it's not that you can't see, because people think hemispatial neglect, okay, neglect, I can't see, I'm not taking any information in. It's the fact that your brain actually fails to pay enough attention to what's coming in. So it's like it can see that, you know, there's let's say you're in a room with a green carpet and you're sat down on a chair. It can see the green carpet, but it knows that, so you know that your leg is there because you know you're born with two legs, but it can't be asked to process the fact that your leg is there. So when you look down, you will not be seeing your leg. So again, disparity on what's affected, for example. So if it's the right hemisphere, you're going to be more affecting your spatial perception. So what's around you, being able to understand what's around you and your memory. Your left hemisphere, again, affects more of your language. So being able to understand what someone is saying to you, what you're saying to them. One funny thing about this is your right-hand side can compensate better for left damage, but not vice versa. So if you damaged your left side, your language side, language is important, but not as important as understanding what's around you and your memory. So your brain has a way of compensating it, forming new connections to be able to relearn language in essence. If you damage your right side, you can't really relearn understanding what's in your environment, and you can't relearn memory. So if your right side was damaged, your left side would be having a very hard time trying to compensate for what's just happened. So this is what happens. This is how they lead lives, and unfortunately they need carers all the time. So hemispatial neglect, they know they have two legs, they just can't see the other one. And what's very common is if you literally take them out to a restaurant to eat food, they will only ever eat half of their plate because that's all they can see. You know, they'll finish this side of the plate and they'll say, okay, yes, I'm done, I'm full. But then all it takes is to literally turn the plate around, and then they see the other side and they say, oh, but I'm not done, but I thought I was done. And that's because their brain hasn't processed the other side. It's only processed one side. If they're drawing, again, you see they've only drawn half of the pictures, half of the face, half of the clock. So yeah, it's a bit detrimental to their lives. They need constant carers because, you know, they'll only end up eating half of their food, only end up, you know, fixing half of their hair, dressing half of their body. It's a bit unfortunate. Another call, try to bring in a little bit of, I guess, psychiatry into this, but oh well. Galastic seizures, which are known as laughing seizures. They look a bit funny, but there's actually nothing, it's really not a laughing matter. It's like any normal seizure, but it's a rare form of seizure. So it's a sudden burst of energy that actually comes out in the form of crying or laughing. And sometimes this laughing can sound a bit more unpleasant, like a malicious laugh rather than a joyful laugh. Now, the damage is caused to the temple. The temple will be behind here, so because you can't see it, it's on an MRI. But and your frontal lobe lesions. That would be the frontal lobe. Remember, parietal, occipital. So tumors, they're basically space-occupying lesions. What tumors they do, they grow and they occupy the space where the brain should be. And remember how I said that the skull is perfectly molded around the brain? If you've got a space-occupying lesion like a tumor, you're taking up the space of the brain. So the brain is actually just squishing upon itself, and it's like, what's going on? Like, you've taken my space, and then causes all these problems. Atrophy, again, when you're losing brain tissue. So imagine like the best way to describe what atrophy looks like is imagine like a full plump grape being, a good brain, and then a raisin being a brain with Alzheimer's. So that would be the best way I can explain atrophy. What happens is going from that grape to that raisin, all crinkly and stuff. Hemiangiomas, I believe they are like, they're not blood bursts, they're like a form of tumor full of blood. Oh, no, it's affecting the vascular system, that's what it is. But normally it's hypothalamic hamartomas, so basically tumors in your hypothalamus. So you can see one literally here, because this is your corpus callosum, okay, a bit more anatomy. And this is your thalamus, and then below your thalamus is your hypothalamus. Again, like the sub, the hypo literally means underneath. So you can see the hypothalamic hamartome here. And again, this is a coronal view. So imagine the crown. This is your corpus callosum. Your thalamus, you can't really see it because we're quite front. So you've basically taken the cut about here, cut the brain through. So you're not going to see the thalamus. The thalamus is a bit more behind, but you can see the tumor here again. So asterignosis, this is something I feel like we take for granted a lot. So if I was to say to you, close your eyes, and I put a coin in your, hand and said, feel this and tell me what it is, you would know immediately that, oh, okay, I've done a bit of feeling. I can feel it's a coin. And that's an asterignosis. It's identifying items purely by touch. So you remember, let's say you left the house going on to school and you put your keys in your pocket. You know that just by putting your hands, let's say you get to the bus stop and you don't know whether you've got your keys or not, just by putting your hand in your pocket and feeling the keys, you know you have the keys. People with asterignosis would have to take the keys out physically and look at them because they can't understand. Their brain does not process the touching into, oh, the keys. That's what they are. So it's unable to identify objects via touch without other sensory inputs, such as auditory or visual. So let's say, I know it was a phone, and you heard your phone, let's say you wanted to call someone, and you put your hand in the pocket, you tried to take out your phone. The person with asterignosis wouldn't know if it was their phone, so it'd have to keep taking everything out of their pocket until they literally look and see that they've taken out their phone. So taking out the keys, taking out a piece of gum, taking out everything one by one until they find their phone, because they cannot identify the objects purely by touch. Even as well, texture, size, spatial properties, so where they are in space, and their temperature. So if you, I don't know, had a nice warm croissant, and you know it was warm because you touched it, but the person with asterignosis wouldn't know because they cannot feel it, cannot feel the heat, have to look at it. Again, this is a lesion in the parietal lobe, so after the frontal lobe. So I know I've run a lot over, but in my defense, the brain is extremely interesting, and if I could go on and talk hours and hours on about the brain. But I hope that this has intrigued you and given you a slight insight into what to expect about the brain, and I feel very, very good thing to go into for research. So I would just like to say as well, join us for tomorrow, the blood, the heart, and the cardiovascular system. My email address is here. Amelia's address is here. This is the website. I hope you've enjoyed today's talk. If you have any questions, please do ask me. I really do apologize for running over, but I hope you have enjoyed yourself. No problem. I hope you guys really, really like this talk. Thank you.